Well, Dave, thank you for that introduction. I'm Matt Sanford. I am the Executive Director of MER, and I am particularly glad to have Dr. John Brockman here with me today. I am not a scientist, and he is, and he's a great educator, and he can really help illuminate some of the topics today better than I can at a high level. And I also can't help but notice, our roadshow about the reactor has been all over. It's been all over the country in the last several months as we try to raise awareness about what we're doing and the need for NextGen MER, and it seems like almost every time we reach an audience, it's after dinner or after lunch. So keep eating. We'll try to keep you engaged the whole time. Also, thank you, so many of you, for being here. As I said, we've worked hard to raise awareness, and I'm curious, how many of you in the room have been to MER and seen the blue glow? About half, maybe a little less than half. That's great. Well, that blue glow that you saw, and those of you who haven't seen it, it's been seen by thousands of people since 1966, when the reactor opened. And the source of neutrons is very special, and it's something everybody in the state of Missouri should be proud about and excited about. So just some basics about the reactor. You can see here, this is a diagram of the reactor from the top. And those of you who have seen the blue glow, that's what it looks like when you're looking down into the reactor pool through 30 feet of water. You can see the blue glow over there on the right and the diagram on the left. It's an innovative design. We were very fortunate back in the mid-50s when this design was chosen. We had no idea that it would be this durable, this versatile, and would have an impact on patients the way it has today, because at the time the reactor was conceived, the idea, the world was just discovering nuclear. We were coming out of this, the dawning of the nuclear age, and we were just thinking about peaceful uses of the atom. The reactor design, not only is it durable, and that really is the reason for the fate of many reactors, by the way, in this country. When I started at the reactor in 1989, there were over 50 reactors at universities in the United States, and now there are less than 20. But this design, in addition to being durable, was powerful. So at 10 megawatts, we are the most powerful reactor at a university in the United States by far, and the only reactor at a university with enough power to make life-saving medical radioisotopes. But perhaps more important than the power is that schedule you see there. There are many research reactors in this country and in the world, and of course many power reactors in the world, and all of them have an operating cycle that has them online for several months and then down for several months for maintenance and refueling. And this reactor is the only one in the entire world that operates 52 weeks per year. 24 hours a day, 6.5 days per week. Now, why 6.5 days per week? That's because on Sunday night at about 10 o'clock, we shut the reactor down every week. And we do maintenance, and we retrieve short-lived medical isotope samples from the reactor itself. And you can think of them like melting ice cubes. A medical isotope decays away. Typical medical isotope decays half of its life in a week. That's one of the important reasons why the reactor operating at 52 weeks is critical. It has to operate every week for patients to receive treatments every week. The other thing I'll say about this slide, you'll see down at the bottom the staff. It is by far the most important thing we have, and that staff and the expertise at MER has been being developed since 1966, passed on from person to person. And I can tell you all that John and I are standing on the shoulders of giants, the people who built the reactor, who maintained it, who trained us to run it and maintain it and to make the most use of it. The staff who run the reactor, who do our research, take care of our safety, everything that happens there, that is one of the main reasons why the University of Missouri and Columbia, we're viewed as the best place for a new reactor to be built. And I don't just say that because we're all here on the Mizzou campus. If you ask people in the world, where do you go to solve tough, complex problems in nuclear science, they say the University of Missouri. Where do we set the example for efficient and safe and reliable reactor operations at a research reactor? It's MUR. Internationally recognized, I think we are more popular in the world and more known in the world than we are in our own hometown. And I'll also say on this slide that we've had an interesting history. So as I mentioned earlier, in the 60s, basic research, what happens when you put something in a reactor? What happens to the material? That happened through the 70s and into the 80s. There was a period in the early 90s when it was even discussed to shut the reactor down. So I mentioned many reactors in this country shut down due to complexities or weaknesses in their design. Others shut down just because they couldn't afford to run. And we had those problems as well. But we had some great leaders, and we were able to change our focus and diversify our focus and stay alive until the 90s. And in the 90s, we really increased our research into medical radioisotopes. That research and those collaborations led to the university pioneering the mass production of lutetium-177. And we'll be talking about lutetium today and how it's used in the latest cancer treatments. We ultimately also developed the ability to produce these isotopes in accordance with FDA requirements. So that's called GMP manufacturing. Those are the controls that go into all the medicines we take. That expertise resides at MUR, where we're able to manufacture active pharmaceutical ingredients that ultimately end up in FDA-approved drugs. So we supplied trials, and we've now also moved into supplying the isotopes for approved drug products that we'll talk about today. So at the University of Missouri, for many years, the focus has been on teaching, research, service, and economic development. And at MUR, we do all of those things, focusing on all four. But you can see there in the middle, research and education, that is the reason MUR exists. And medical isotope production is very important to patients. It's important to MUR, but it is not the only thing we do, and it's really not our only vision for the future. So when we talk about NextGen MUR, a new reactor, of course, it will produce more medical isotopes. But when John speaks later, the research programs, the education programs we have will also be expanded. So you've been to MUR, many of you, and even if you haven't, I'm sure you've heard about our supply of medical isotopes and how we are really the only supply for several key isotopes in the Western Hemisphere or in North America. Those first two listed, Lutathera and Pluvicto, those use lutetium-177. That's the active ingredient provided by MUR. And John will talk in a moment about those technologies, but I'll say that they're making a difference in patients' lives. In some cases, saving lives, in many cases, extending lives, and improving the quality of life for people who are impacted by cancer. The case of Lutathera, which is used for a kind of pancreatic cancer, 72% reduction in disease progression or death in the data. Pluvicto, also newly approved, incredible PSA response from patients treated with Pluvicto. TheraSphere. So TheraSphere was developed in partnership with scientists at the University of Missouri-Rolla, or S&T, ultimately commercialized. So that is a treatment for primary liver cancer. And 93% of patients receiving TheraSphere have an increased overall survival past 3 years. It makes a difference in their lives. Iodine-131. Very common cancer, thyroid cancer. It's one of the leading cancers on the rise in women. Iodine-131 is a near cure for thyroid cancer. 98% of patients receiving iodine are cured, and myrrh is the only producer of iodine in the Western Hemisphere. So the number of patients is incredible. So now it's about 500,000 per year. That is the stadium sold out for every home game in a season, just to give you a feel for the scale of patients impacted by isotopes produced at myrrh. And I'll give you one specific example. This is Steve Reed, and he was a geologist at NASA. Steve Reed was diagnosed with prostate cancer in 1998. And since that time, he went through 18 rounds of chemotherapy almost annually. His quality of life was not good. A couple of years ago, he had really reached the end of the rope, and he had reached the end of those treatments, and he met with his doctor who suggested Pluvicto, and he received the treatment, which is administered by IV. And his PSA levels went from the 200s to 5. But most importantly, he was able to continue his quality of life, which had really been tough for almost 20 years. And he was able to resume normal activities. So it's every day we see stories like this, and it's why we do what we do. With that, I'd like to introduce Dr. Brockman, and we'll trade off a little bit for the rest of the slideshow.
Thank you, Matt. Can everybody hear me? Great. Okay. So I have the pleasure of working at the University of Missouri Research Reactor as the Associate Director of Research and Education. I've been there for some time. I actually was a postdoc at the reactor back in 2006 to 2008 and managed to beg, borrow, and steal various positions until I find myself in this incredible position that I'm in. And it's a pleasure to talk to you all about the research and education programs that we have going on. I think we're going to start with a video. Maybe some of you have seen this before. It's played at most of the home games for basketball and MU football.
From its proud home in Columbia, Missouri, the University of Missouri is one of the most important and reliable producers of medical isotopes for treating cancer in the United States. The University of Missouri Research Reactor, known as myrrh, is the only U.S. producer of 4 critical medical isotopes. Researchers at myrrh create radioisotopes that are used to diagnose cancers and heart disease, and in the treatment of liver, thyroid, pancreatic, and prostate cancers. These are life-saving treatments that those with cancer desperately need. Myrrh's central location and unique capabilities ensure rapid transportation of radioisotopes to more than 1.6 million patients a year across the nation and beyond. The demand for radioisotopes is rapidly increasing, however, and work must begin on a new, larger reactor, NextGen myrrh. NextGen myrrh will solidify the University of Missouri's position as one of the most important sources of radioisotopes, while expanding critical cancer-fighting research and medical isotope production for generations to come. Without a new reactor, these critical radioisotopes may not be available in the U.S. in the future, leading to dependency on foreign sources. Despite their minute size, radioisotopes create massive opportunities in the treatment and imaging of numerous cancers and diseases. But how do they work? A radioactive compound produced at myrrh is linked to a targeting molecule that seeks out certain proteins on a tumor. The compound, linker, and targeting molecule form a radiopharmaceutical. Cutting-edge science and medicine meet when the radiopharmaceutical is administered into the body and sticks to cancer cells. The radioisotopes release energy that acts as a wrecking ball to cancer cells, eventually destroying them while leaving healthy cells unaffected. As a result of this advanced treatment, patients have the ability to resume a vibrant and healthy life. For more than 50 years, myrrh has operated safely and has been at the center of advancements in science and medicine. NextGen myrrh will take us further into the future, providing accessible and life-saving innovation for generations.
How many have seen that video before? That's great. That means we're getting the word out. So I'm going to spend just a little bit of time talking about this targeted radiotherapy approach. The difference between this approach and how we have previously thought about treating cancer with radiation is that it's not an external beam therapy. In an external beam therapy, the radiation is generated outside of the patient and then is pushed through. In this new type of therapy, or targeted radiotherapy, the radiation is found in a molecule that's internalized in the body. And you can see that molecule right there, the radioisotope. So that's what makes the MURR special. We are a major source of that radioisotope. And in fact, we're a major source of a particular kind of radioisotope. So that radioisotope that we make at the reactor, we make it by irradiating it with neutrons. And when you add neutron to an element, you make it radioactive and it becomes what we call a beta emitter. Has anybody ever heard of a beta emitter? Does anybody not know what a beta emitter is? Okay. So a beta particle is just a fancy name for an electron. Remember what an electron is. It's specifically an electron that's made in the nucleus. It comes out of the nucleus of the atom, not the cell, made in the nucleus of the atom, and it comes out with high kinetic energy. That's what we make at the reactor, is these beta-emitting radioisotopes. And those electrons are then capable of moving through tissue up to several millimeters, and as they move through tissue, they cause damage, mostly single-cell DNA breaks. And that ultimately is what kills the cancer cell. So in order to make this therapy work, we have to have the radioisotope, but that's not all we have to have. So there's a part of this, it's called a bifunctional chelator, that holds the radioisotope. That's the purple part. That's the chelator and the linker. Okay, so we have some chemists that now come into the picture. And these chemists, they design molecules. The point of the molecule is to hold the radioisotope very tightly. We don't want it to let it go in the body. We want it to go where we want it to go. We don't want it to get out, right? So chemists are working on that problem. And then in order to make this into a smart bomb, they attach that chelator and linker to a ligand. And that ligand, it could be lots of different things. It could be a small molecule. That's PSMA 617. That's in Pluvicto. It could be a peptide. That's dotatate. That's in Lutathera for treating neuroendocrine tumors. Or it could be a monoclonal antibody, or any number of varieties of these things. So the sky's sort of the limit on how you create these ligands. The point of the ligand is that it goes to a part of this tumor cell that overexpresses some antigen, some target, right? And so when you deliver this whole conjugate, this whole bifunctional chelator with the radioisotope, it selectively then goes to these sites in the tumor cell which are overexpressed and delivers this radiation. Now, the beta particles have another advantage. So I told you that they have path lengths of several millimeters. They go through many cells before they lose their energy and become less potent. And that's advantageous because in a person who has cancer, they often have many different types of tumor cells, and not all of them may express this target. However, if they're in the same area as cells that are expressing this target, then the beta-emitting particle will also move through those cells and affect cell kill. So one of the advantages of this technique is you deliver this radiation specifically to the tumor cell, and the result is many, many less side effects. And you hear that over and over again with Pluvicto and Lutathera patients. It's much less hard on the body than chemotherapy agents are. And you see this quote here from a nuclear medicine physician from the University of Washington. The greatest strengths of these treatments come from what the patients are telling us. It improves their quality of life rather than causing it to decline. So there's another part to this story. Some of you may have heard the term theranostic. I've told you about the therapy part of the molecule, but we can also create molecules that we use for imaging. So This shows a theranostic pairing. And what you see here is dotatate in the middle, and you can label it with two different radioactive atoms. The gallium-68 releases a different kind of radiation. They're called positrons, and positrons create two 511 kiloelectron volt photons that go in opposite directions. And so you can image that. That's called a PET scan. So we can place the patient into a PET scanner, and you can look and see that they have the target that this therapy agent is going to go for, right? So in this case, it's a neuroendocrine tumor. You can see in this PET scan that the gallium-68 is showing that it's going to these neuroendocrine tumors. So you see what you treat. And after you visualize this, you've demonstrated that the patient is going to respond to this through the image, you know that the therapy is going to work. So you see what you treat, and then you treat what you see. So the way that you treat is you use the same molecule. So there's the molecule in the middle again, dotatate. And instead of labeling it with gallium-68, it's labeled with lutetium-177. And that's Lutathera. So now we're treating the very same tumors that we just imaged. Okay. This is called a theranostic pairing. And it's an advantage that this particular kind of cancer treatment has over immunotherapy, over many other types of therapies, right? Because in those other therapies, you wait for a patient response before you know if you worked. Here, you know going in it's going to work, and furthermore, you can image after the treatment, and you can see, do I need another treatment or not? How am I going to change the treatment plan now that I know what the result of the last treatment was, right? So this is the power of theranostic treatments. So back out again to look at the University of Missouri. So Matt has told you about how the University of Missouri is unique because we have a 10 megawatt research reactor. It's a very powerful research reactor that's very good at creating high levels of beta emitting isotopes that you need for these therapies. But it's sort of like, that's not all, right? We have also, for 60 years, had a very deep bench of radiochemists, and they've been spread out all over campus. So they've been in the chemistry department, the biochemistry department, at radiology, at vet med. And in the last couple of years, we have made an effort to get all of these people together, to give them a place to congregate, and to give incredible state-of-the-art laboratory facilities to do the basic science. And so that's the Molecular Imaging and Theranostic Center. There is not another place like MITSI in the United States. It has some of the most impressive laboratory space and small animal and synthesis capabilities that you will find anywhere. In addition to that, we have the College of Veterinary Medicine, which has long been a collaborator with MUR and the chemists that we have. Some of the very first work that was done on Therasphere and Samarium-153, which was used in a Quadramet, another commercialized product that came from the University of Missouri. Those studies were translated at the College of Veterinary Medicine, right? And they have the capability to do companion animal studies, which is very unique. There are not very many other veterinary programs in the United States that could come alongside the reactor and do those kinds of studies. And then the campus is further distinguished because not only do we have a reactor and a MITSI and a College of Veterinary Medicine, but we also have a medical school and a cancer center here. And so work is going on now to work with the Cancer Center and the NextGen Precision Health Center to explore first-in-human kinds of studies with new radiotherapy agents. It's a very exciting time. And I want to tell you just a little bit more about how all of these pieces can work together to achieve something really amazing. And I'm going to show that through an industry collaboration. So ABK Medical is a company that came to the University of Missouri to collaborate back in, well, they started in about 2012. They probably came to the reactor around 2016, 2017. They came to the reactor because they needed isotopes. They needed an isotope supply. And they had a great idea. Their idea was to make these, they call them I-90 microspheres. An I-90 microsphere is a radioembolism technique. It's a little glass sphere that's tuned to deliver radiation dose to the liver, just like a thyrosphere. But what distinguishes it is that it's radio opaque, so you can image it as the interventional radiologist is delivering them into the tumor. And they needed lots of help. They needed help from the reactor, they needed isotopes, but they also needed lots of other things that we could provide, like developmental radiochemistry and QC testing of products and expertise in handling high levels of radioactivity, processing, and shipping. Shipping is one of our superpowers, by the way. And they needed weekly batches of isotopes to develop their product and ultimately go for a clinical trial. But that's not all they needed. So they came also with a dosimetry problem. They did not understand well enough how to do the dosimetry for their product. And we were able to pair them with Jeff Bryan at the College of Veterinary Medicine, and they were able to work through those issues as well. So what's really exciting about this story is the next step, which is a partnership with MU Healthcare to do a first-in-human or a clinical trial of their device, and Dr. Ryan Davis is right now leading that particular study, and we're waiting to enroll our first patient. This is a very, very exciting time. It showcases how all of these parts of the University of Missouri can work together towards a shared purpose. And I've talked a little bit about beta emitters, and lutetium-177 is the one that we make at MUR in large quantities, but there's lots of exciting potential for the future. So beta emitters, they have this long path length. They can treat tumors that are large and bulky. They move millimeters through tissue. But there's other isotopes that we might be interested in as well, other forms of decay. So one of those is an alpha emitter. Instead of an electron, an alpha particle is a high energy helium nucleus moving through space. And it deposits its energy very, very quickly in a very short amount of space. It causes double-strand DNA breaks instead of single-strand DNA breaks. It's highly efficient at killing cells, and it has a very short path length. So think 1 to 2, maybe 3 cell diameters. There's lots of clinical trials now going on in the alpha emitter space, and MITSI is also working on basic and translated research on several alpha emitters. And then another exciting possibility is Auger emitters. So I've listed here terbium-161. Terbium-161 is an isotope like lutetium-177 in that they both have the same half-life. They both have the same energy of beta particle, electron that they release. But terbium is also an Auger emitter. And an Auger electron is a very low energy electron. It has a very short path length, like it won't even cross a cell, right? Very short. But it also deposits a lot of energy in a very short amount of space. And so there are a lot of people interested today in if you replace lutetium-177 with terbium-161 and you have these Auger electrons, does that give you enhanced power to kill individual cells? And the early work looks very promising. And I'm also proud to say that the University of Missouri has been the tip of the spear for terbium-161 research in the United States. Our own Heather Hinkins had one of the first DOE isotope production grants to make this and was used in early work at MITSI. And so, the future is bright for radiotherapy research at the University of Missouri. And Matt, I think I'm handing it back to you now.
Thank you, John. So what an incredible opportunity for patients. And what does this mean for the University of Missouri, and where do we go from here now that there has been this awakening? Carol and Anderson, you're in this audience right now. I think we don't like to use the word renaissance that's happening in our industry right now. I'm not even sure why, but it's an awakening. And these discoveries have led to a tremendous amount of activity. Multiple companies have been formed. multiple billion-dollar-plus acquisitions, companies pursuing these technologies. How can we expand what has already been started? Now this diagram, it does not matter that you can't read the specific names of the companies around the outside. If you look at the outside, those are many new companies, some existing. And if you look in the very center, you'll see Lutathera and Pluvicto. Now those are the only two drugs using the technology John just described, the only two that are approved by the FDA. In the middle and all the different rings, those represent clinical trials using different isotopes, targeting different cancers. And they're all marching toward the center. And many of these companies have great ideas, and we think many of these technologies have the potential to reach many more patients and attack many more cancers. But most of these have something in common. So they all need access to the radioisotopes, and they need help developing these technologies. And that's the expertise we have here at the University of Missouri, and that's why we see such a need and why we see an opportunity to apply our expertise. So this picture is happening at MURR. There's a lot of confusion around MURR right now, so how many of you have heard of the NextGen MURR reactor? Almost everybody. That's good. That's good. How about MURR West? Right. So that's not the same as the new reactor. MURR West is a building addition we're used to expand our research and our industry partnerships. The building in the picture here has not been announced. So that's being built in cooperation with industry to expand radioisotope production. And I'll tell you something about this. So this allows us to make more of what we're making right now because the demand is so high. And one of the reasons we're even able to make what we have now is we've set up temporary labs that will move into this new and more modern space. And setting up that temporary lab is a huge undertaking by the people at the University of Missouri, all the way to planning, design, and construction. We have people on the ground today in Chicago loading a new hot cell, over 100 tons in one hot cell line to manufacture one batch of NCA lutetium. Those trucks will be arriving tomorrow, and over the weekend we'll be staging hundreds of crates. And then on Monday, installers from Italy, where the hot cells were built, will arrive and start putting those hot cells together in this temporary space over the next 4 months. And then in October, we'll launch another expansion. This building you're seeing here is for expansion beyond that. And then here in this drawing, you see off to the left that taller building. That is another one we've just started. And I think the university this fall will have a groundbreaking to better explain these buildings. But just to give you this, a sense for the scale of growth that's happening and what the need is and how industry investment is pouring into the university to help make this happen. And it is a sign of the potential to grow. Now, this building, lots of confusion around this. This is a drawing of what we call the Radioisotope Science Center, which is not certain to happen. It's really important that I make that clear. But we mentioned companies being formed. Well, there is also the Department of Energy. They have reactors all around the country. Great reactors, much bigger than ours. But the idea is that the Department of Energy is also interested in making isotopes that can't be made by industry for various reasons. They need help with the actual handling. The expertise at the University of Missouri really is unparalleled in this country. And so the idea for the Radioisotope Science Center is this would be located at Discovery Ridge. And it would be a place where the Department of Energy can bring isotopes in, either from MURR or even from other reactors such as Oak Ridge National Laboratory, for people at EMU to handle, do the radiochemical processing, and help distribute them in the country where they need to go. Now, this is pending a grant from the Department of Energy. We do have $20 million from the state of Missouri that would be a 50% match to make this project happen. So fingers crossed on the DOE grant. If that happens, we'll make announcements, and then ultimately we'll start construction of that building. But it's a great and exciting opportunity for the university. And then NextGen MER. Well, there's a problem if you go back to this slide here. The two isotopes in the middle that are approved, Lutathera and Pluvicto, I wouldn't say that they have the MER reactor full, but they have certainly taken up a lot of space in the reactor. That's 2 approved drugs. What happens when there's 20 different isotopes in addition to lutetium? This isn't an issue just for us. This is an issue for patients across this country, that we may have new technologies that can save lives, extend lives, improve the quality of life. The science discovered, figured out, we know what to do, but we can't make the radioactive material. Can you imagine? And we think the University of Missouri can be a part of that solution, and that's one of the driving forces behind the NextGen MER idea. So this reactor would be more powerful, but it would also be physically larger and have more payload. And I think it's important to talk about scale for just a minute. So when you think about 500,000 patients, do you imagine truckloads of material leaving the reactor every day? Most people do. It is not like that. Now, big science happening at MER all the time. With the things we irradiate in the reactor, we put in what we call cans. Our terms are not all that scientific sometimes. Inside cans in the reactor, we have vials. And I realize you can't see this vial very well. It's small. And in the end of this vial is a tiny bit of colored material that we call a target. It's that gets put into the reactor that ultimately becomes a large batch of material that can be used to make the treatments for hundreds of patients. So the amount of material that we're talking about here is very small in physical size. These cans stack in the reactor in an area about the size of my forearm. That's it. That's where your world supply for some isotopes comes from. It's where the nation's supply comes from. The point is, it's limited. And who knows what will be discovered next. So we have the expertise. We're certainly in need, and that's why the president of the university and the board of curators have prioritized making this project happen. I think even today, the university issued a release thanking the state of Missouri for a $50 million appropriation to help make that reactor project happen. And I'm sure there will be questions in the Q&A about the reactor. But first, let's watch Nicole's story.
6 months before my diagnosis, I was just a normal 32-year-old girl. I was happy and really just was enjoying life. The first prognosis that I received wasn't good. We were just in complete shock. I had about 6 months to live. My tumor was inoperable, and there was really nothing that they could do for me, like what just happened to me today. I didn't want to just succumb to that. I wanted to get another opinion, and when I went to meet with them, they wanted me to test out this other therapy, TheraSphere. Dr. Baker is the director of the liver tumor clinic. She believed enough in herself and her team's abilities to be able to take on a case like mine. And the best I could do was try.
We are able to give high dosage of radiation to a tumor to affect kill, and also give lower doses to the liver surrounding it, which has changed what we can do as surgeons to facilitate options for surgical resection.
I was obviously very scared. I didn't really know what to think. I had enough faith in that interventional radiology team to help me get better. When we found out that the TheraSphere procedure worked, that was one of the happiest days of my life.
When I reviewed the scans, my initial response was that it had done exactly what I needed to happen. There was no progression of disease, and we could have many options when we got to the operating room. And that's why having these relationships with our medical oncologists, our hepatologists, and our IO people is essential to be able to get the patient the best outcome.
It was because of her team and this technology I'm here today. If it was the start of a second chance for me and I'm healthy, TheraSphere saved my life.
As we kick off the question section, can you talk a little bit more on current clinical trials?
So there are clinical trials ongoing right now. The couple that I'd like to highlight is one on advanced melanoma, phase 1 and 2 trial using lead-212. There's multiple prostate cancer trials going on. Those are either to move the Pluvicto closer to the start of treatment, or to check another isotope like actinium-225 or terbium-161. Then there's clinical trials in other space too, involving breast cancer using a variety of different isotopes.
Don't wait for the mic.
It's off, so whatever.
Okay.
So you said a lot of this is kind of, we got radio pharmaceuticals. Between the two reactors, how do we see the kind of the division of education and research being split up then? Will it be mostly here on the existing MER or the NextGen, or how do we kind of partition that?
Yep, that's a great question. A common question is, will we operate both reactors when the new one is built? And the short answer is that yes, that's the intention. The current MER reactor is licensed through 2027 and through 37. Thank you, John, 37. And through recent regulatory changes, really, there is no foreseeable end to its licensed life and its functional life. We don't see any limits there either. So it's really indefinite that the MER reactor would run. Now, the NextGen MER reactor project is in what we call preliminary design. That will then lead to detailed design, and those design processes lead to engagements with the Nuclear Regulatory Commission that ultimately lead to licensing and a construction permit, and only after you receive a construction permit can you put a shovel in the ground and start working on the reactor. And the reason I say that, to answer your question, is in the preliminary design process, all of the facilities at MER that are used for research and medical isotope production are evaluated. Which ones make sense to reestablish at the new site at Discovery Ridge next to the reactor? Which ones make sense to keep using at the current site? So the split of isotope production and research, where those things happen, is yet to be determined. Great question.
These isotopes, you know, a long time ago, I worked with I-125 in the lab, and we did lab tests. And we had to dispose of I-125 because, it was a radioisotope. So when these isotopes are in someone and they're targeting these cells, do they go eventually to a waste product, or do they completely deteriorate so that they are in, like, they're not reactive any longer?
So the isotopes that we use for imaging tend to have shorter half-lives. They go away pretty quickly. So you give them, and maybe as short as a day later, there's no detectable activity. Could be longer. For therapy isotopes, they tend to have longer half-lives. So 6 days, maybe, for lutetium-177 and terbium-161. And there you do have to worry a little bit more about what the patient's doing and where the patient's going after the treatment. Now, the regulations, I think, say that after you've put it into a patient, into a person, then their excreta, right, that we don't have to worry about that radioactivity. But in reality, we do. We don't want them, right, to put others in jeopardy or anything like that. And so there's a lot of work that goes in on the back end to make sure that we have appropriate patient care and training about where to use the facilities and how to clean up and that sort of thing.
One more question.
Yes.
So when somebody has, like, that patient with liver cancer, did she have to go through chemo and other treatment before she could ever get to these clinical trials? Did she have to go through all that hard part before she got to this?
Yeah, in that particular case, that wasn't a clinical trial. That's an approved drug product. And I don't actually know if she was treated with chemotherapy before she was treated with, Jeff is saying no, he probably knows, right? And so in that particular case, the patient was just treated with TheraSphere, and then after the treatment and the tumor debulked, they were able to then do a successful surgical resection.
With the resources that you're providing, do you have enough medical teams in this area to use these?
Yeah, that's a great question, and that's one that's being confronted by our clinical trials task force here at MU. We're actually fortunate today to have Dr. Rick Barron in the room with us, who leads our hospital system, and he tackles those issues, the one you just asked about. So increasing the number of trials and treatments using these materials in the hospital, it is a challenge. It has to be managed. Anyway, to your point, wait, what was the second question? Give me the second question again. The one you just asked.
Just the resources. Do we have the resources in this area?
Oh, right, sorry. So we do see a gap there in the number of people that we have that are needed to make this happen. And our task force today is focused on identifying the additional space we need, the people we need, and the equipment. We see some gaps in all of those areas, but fortunately, there are gaps we think we can fill. So no, we don't have everything we need there.
Okay, thank you. One more quick question.
Yeah.
These isotopes have a really short half-life, correct?
Yep.
So what is the most expeditious way, and what do you use to get them to their destinations?
Yeah, the logistics for the different isotopes vary. You'd be surprised. In most cases, the idea to pursue a new isotope treatment doesn't get very far if the logistics don't work, and that's reviewed early. But the supply chain, where the isotopes are made, how far they need to go, and how they get there, is a big part of what we do every day. Typically, an isotope is produced quickly at the reactor into the form needed by a pharmaceutical company. It's often then shipped by truck, and in many cases on the same trucks you would see with your Amazon products. So these are very low-level radioactive materials, heavily shielded by lead in some cases, sometimes other materials. Typically that's enough time to get the isotope in bulk where the drugs are formulated. Then they're shipped to radiopharmacies in some cases, which are near hospitals, and then they get into the patients. But the exact logistics depend. These range from days to weeks, the half-life for these different isotopes. Some minutes in the case of imaging isotopes, or hours.
So do you see this technology kind of going to liquid tumors, or tumors that are quote-unquote hard to reach, like brain tumors or vestibular schwannomas or places where basically there are no other options than chemotherapy?
So I think it depends on the tumor itself, right? So this targeted radiotherapy does give you a way to go after some of these dispersed tumors, right, that are in the body that aren't solid tumors, but you may have to change the isotope that you want to use when you do that. So it might be lutetium-177, which has this long path length, may not be the correct choice. You might be designing new agents that use lead-212 or one of the Auger-emitting isotopes.
So your graphic was very impressive. We have a tsunami of studies coming to clinical trials, and supply clearly is the big gap. Are there other areas across the country and world that are trying to fill this gap as well as University of Missouri? And how much of that gap do you think we will be able to fill? If all those studies go to clinical trials, it's a non-linear rate. It's actually very, very impressive, but maybe theoretically scary if we're close to capacity. So do you think we can fill that gap?
So there are very few reactors in the world right now that can do what needs to be done. Our competition is mostly in Europe, but many of those isotopes can be shipped. The half-life around a week is long enough for those logistics to work. But I think the world eventually solves its problems. I think it's recognized this need. We aren't the only one that sees it. And I think what we'll see over time is the need for radioactive materials be met. I think we see right now power reactors in the world converting some of their facilities to be able to make medical isotopes. MUR is the only project of its kind that I know of in the United States. But I think the real gap will be in the development area. The companies, even the big pharmaceutical companies who are getting into this, don't know what they don't know. And when it comes to developing the technology, that's one thing, but scaling it up is another. And I think that's really where the University of Missouri will shine, is helping bring the ideas to reality and having the reactor close to where the clinical trials can happen and where the radiochemistry can be developed, I think will be a real key. And I don't see a lot of people answering that call. I do see the reactor problem being solved eventually.
Great. For the sake of time, if there's no other questions, I would like to thank you both for the work you're doing to innovate here at the University of Missouri and have impact on health here in the state of Missouri and beyond. And for that, I would love to share a token of our appreciation to you both. They're personalized, so I have to make sure I get this matched right. So here for you.
Thank you.
No.
So thank you so much for sharing your work and the work that you do. Let's give one more round of applause. And please join us next month, July 22nd, either in Kansas City or online. Thank you.